tomorrow there is a variant also, we should know how the the innate immunity cells are produced, what is happening during infection is an important area to talk about. And I said in statistics, both these names are very well known, and we have had a lot of philanthropists who have helped us to do it. Now, when the Department of Biotechnology was set up in 1986, and I was one of the early first faculties to join this program. The focus was at that time the visionary in Dr. Ramachandran, who was secretary then. He started the PG programs. We had the microbial culture facility, animal tissue culture facility, and plant tissue culture facility. The National Animal Tissue Culture Facility came up in Pune under the leadership of the late Ullas Wagh, who did a tremendous job to keep on collecting the enough cell lines all over the world, so that scientists could grow, because these cell lines are required for growing viruses. If NIV has helped us to make the first COVID vaccine, it is because we need some cell culture facility or some way to grow the virus. And this was done originally at NIV, then the National Animal Facility for Animal Tissue Culture Collection was another one, and plant tissue culture was the other area which came up. Okay. Now, I just want to quickly go through that. Everybody is now talking of translational research, and what I want you to know that I was fortunate to work once with a company called Span Group of Companies, which was the ideal environment where we started the MSc PhD program in clinical biochemistry. I am talking of way back in 88. So, I think industry should also have an academic program, because universities alone cannot cater to the how to make a product. The way we talk about product development, we need parties to help us out, and no institution, no IIT can create a product without having linkage with various things. And I remember in India testing was a big problem. So, when I was general manager in Ranbaxy, I brought in the specialty Ranbaxy laboratory concept, and today you can do any testing due to this. So, I think we need also visionaries who think for the future of the country, as Mr. Love Agrawal beautifully put it, that all of us have to join hands to run the country, get over the problem. And I think I would like to tell you that at that time in 94, I developed a HIV kit for Ranbaxy, which was less than 2 rupees a test. So, I think it is possible because of right collaborations which happen. So, industry alone cannot do it, academics cannot do it alone, and you also need regulatory agencies. So, so any program should teach them all the factors responsible, not just teach the science. That is why I use the word life science is not really the right word. If I were there to choose, I will use some other word, just for that is my personal view. You can accept, you can reject it, no problem. Okay. And then of course, everybody is now talking of long distance. When I was vice chancellor of Nagaland University, which is a very remote area in the country of the Central University, we were the first to bring in long distance, what we call, we were the first to implement NPTEL program, a national program or technology enhanced learning. It was there, but nobody gave a value to the whole effort which went in. I am talking of 2008. So, we were the first to implement, we put optical fiber, and you won't believe, even we started telemedicine those days. So, it is not that COVID brought this teaching to a halt, it is wrong, because we were well suited to do that. And I can tell you from experience that remote areas in India needs lot of hand holding, and this kind of IT related service is very, very important. I think it should be continued, because that becomes easy to network, monitor, etc. Okay. Now, the hottest area of course is the genomics and clinical research. So, that is a hot area which is happening. I am sure a lot of people are going to talk about it, and I want to say now this is going to be the growth driver. Many of the old diagnostic tests are all getting into the vaccine, because now we can have much more power. So, it is genome is very important, technology is important, awareness is important, it is also affordable. So, I think, and I must say some of the biotechnology students have set up companies and which are doing amazing work in the country. So, I think a lot of startup companies and some of the established companies, a lot of youngsters, not the big companies, who have made this growth driver possible. And many of them have been associated with me. So, it is a pleasure to use their slides freely because I have a direct relationship with them. So, the major thing is preventive, predictive, personal and precision medicine is going to become a $5 billion industry, and market for genomic basis doubling every year. And the diagnostic market, they say it is about 68,000 crore or whatever it is, but currently it is valued at about 50,000, but some of the areas are growing, which I am sure the business people will talk about. And another area which has come up is called protein targeted chimeras. I think this is a very hot area, in the sense that sometimes there is no destruction of protein which should have happened in the body. So, there is a whole new technology which I used to work on it about 30 years ago, where I used to work on ubiquitin, and that same system, I do not want to go into details of it, but today they have become targets for solving a lot of disease problems. Because of paucity of time, I would not spend much, but I want you to remember this word. Please go and search in the net what is PROTAC, and it is just in the nascent field, and we are able to do some of the cancer treatments, etc. Might be solutions are going to happen through this, because they are otherwise very difficult to target. I just want to quickly go through that. All of you should be aware that the CSIR was the first one which was created. Post independence, we created ICIR, ICMR, Department of Atomic Energy, DRDO, etc. All these agencies are there. So, the government has been, or the policy makers have been very good. And just to give an example, because I did my PhD from CFTRA, we are always proud that CFTRA made the technology for making baby food, and thanks to Verghese Kurian and NRDC, they did even clinical trial in in the 50s at the Vellore Institute, Vellore Medical College, and then today India is free from import of it. I think many things will have to be free from it. So, I think this is a beautiful example of baby food production, which has saved a lot of lives of children, the infants, etc. So, a lot of interesting things have happened. So, what my last message is: inspired by coming together, ideate by keeping together, and implement working together. Thank you. Yeah, it's over to you.
Thank you, sir. Thank you, sir. And once again, we apologize for the glitch
No,
that.
no, please don't apologize. It is beyond your control. Please don't do that.
Thank you so much. Thank you. Now, may I request Dr. Shweta Singh ma'am to deliver the vote of thanks to all our guests.
Thank you, Vaishali. Good morning once again to one and all present on this meeting today. Honorable Chief Guest, Sri Lavagarwalji, respected guests of honor, and all the invited keynote speakers and guest speakers and participants. It is such an honor for me to get the opportunity to thank you, all dignitaries. On behalf of CIR, I extend a warm welcome to the people in the gathering. I would like to express my gratitude, all the esteemed authors and participants of the conference for their presence and contribution to make this event a great success. I extend my gratitude to our Honorable Chief Guest, Sri Lavagarwal sir, to take out time from his busy schedule to grace the event. Thank you for inspiring, encouraging with your words on this special day. A special thanks to Shri Kumar Shekhar Vijendra ji sir for making Shobhit University the academic partner for this conference and providing all the support possible to make this event successful. My sincere thanks to Dr. Raj Narayan Tiwari, Director ICMR, Professor Kanan from KK Biotech to grace the occasion by their presence. Also, I certainly believe that attendees of this conference will learn a lot from the talk of Dr. Ashwini Kumar from IIT Kanpur and Dr. Devnath Pal from IIC Bangalore in the subsequent technical sessions. Finally, I welcome all the speakers for the panel discussion, which is going to be held later in the day. I extend my gratitude to Professor R.M. Mehra and my team, CIIR, for organizing this conference with a lot of enthusiasm and passion. Thank you once again.
Thank you, ma'am. Further, I would like to help please accept our token of appreciation from Team CIR to all the eminent speakers.
Thank you.
Now, I welcome you all to the session 1 of the conference. May I request Dr. R.N. Bharal to become the session chair of this session. Everyone, we have with us Dr. Ashwini Kumar Thakur, Professor, Department of Biological Sciences and Bioengineering, IIT Kanpur, who will be obtained his PhD from Institute of Microbial Technology and Punjab University, Chandigarh. He did his postdoctoral work at University in Tennessee Medical Center and Pittsburgh Medical School. He is a recipient of Alexander von Humboldt Fellowship and worked at Max Planck Institute of Biochemistry. Prior to joining IIT Kanpur, he worked as a senior manager in Biologics Development Center, Dr. Reddy's Laboratories. He joined BSBE on 6 May 2010. Another area of interest is to utilize self-assembly of protein in material sciences and bioengineering applications. Sir, you can take it forward from here.
Am I audible?
Yes, sir. Yes, sir.
Okay. Thank you, Vaishali, and thank you, Himanshi, for giving me this opportunity to come to this conference. I would like to, can you please enable me to share the presentation? Okay, so my slides are visible?
Yes, sir, they are visible.
Okay, thank you. So thank you organizers for giving me this opportunity to present my laboratory work in the second National Conference of Engineering, Arts, Science and Technology, where you have put a nice theme on life sciences and healthcare management. I would like to thank CIR and Shobit University, Shobit Institute of Engineering and Technology for giving this opportunity. I will not talk about healthcare management per se, but would like to bring on some of the product discoveries which have originated from our fundamental research in protein, peptide, and understanding of amino acid self-assembly. So before going into my talk, I would like to introduce my department at IIT Kanpur. BSB was established in 2002, and we are working in 4 areas of biological sciences and bioengineering. The first one is molecular, cellular, and developmental biology. The second one is structural and computational biology. The third one is bioengineering, and the fourth one is neuroscience. And we have a separate department now on cognitive sciences. So this is for the students. If they are interested, they can join BSB department of IIT Kanpur. And in another development, we are going to have now a department, a center for engineering in medicine, which is going to be very unique in the whole country and probably in the world, where engineering and medicine with biological sciences is going to be practiced to develop cutting edge technologies in medical and healthcare sector. So in this center, we are going to look forward for faculty members and students in the areas of regenerative medicine, molecular medicine, and digital medicine. Professor Kanan has very nicely put a lot of positivity that how India can build further and bring scientists, technologists, and industrial people together to cater to the needs of healthcare in India. So our biological sciences and engineering in IIT Kanpur is also working very proactively in these particular areas. In addition to this center, we are also going to have now a super specialty hospital, which will be also going to be very unique, where technical education, medical education, and bioengineering education is going to be integral part of training of engineers and next generation medicals. So if students are interested, I would like to share that how we admit in our department students of various streams. So we have BTech, MTech, BTech, MTech dual degree program, BTech double major and minor programs in our department. We admit students for PhD, BTech of any branch, not essentially biotechnology or biomedical engineering can apply. BTech of any branch can apply to our department. MTech of any branch can apply to our department. MSc, any branch of life sciences, agriculture, MBBS, BVSc, pharmacy can apply for PhD program in our department. And we also have a provision for extra degree. For example, if a student arrives for a PhD degree, they can also get an additional degree of MTech if they prove to be exceptional and carry out multiple projects. And in IIT Kanpur, we also have an institute postdoctoral fellowship, which is also very unique in the country as of now. So I request students to explore BSB department of IIT Kanpur, and one can write to different faculty members of their interest and or visit our department. So with this short introduction to the department, I would like to go into the main talk of today, where I am showing a pictorial representation of a protein, which all of us are aware of, that our cell and body works because of the proteins, which are generally spoken to be well folded in nature, and then only they function. Okay, and it is also known and calculated now how many proteins are present in a particular organism, so as to carry out all the homeostatic and metabolic activities of the body. However, in many instances, this folding and structural adaptation does not take place. And in very limited cases, it is possible that protein will undergo different steps and stages, and eventually they will become long fibrous structures. Okay, and slowly and gradually they will build in tissues and organs to make these fibrous structures very long, more in amount, and simply they are capable of choking a particular organ tissue, and sometimes involvement of multiple organs in the body, and leads to uh, a, a disease which is known as amyloidosis. And there are around 40 proteins which are known to make such fibrous structures in human as well as animal bodies. Okay, so in another very exciting area, these proteins can be also allowed to form spontaneously in a reaction vessel or in test tube, due to their inherent tendency to not stay in the native state, but to transform into the fibrous state, as I am representing here. You can appreciate the fibrous structure of this. And what people have found, that such fibrous structures are having very good mechanical strength, which is at par with, let's say, steel. And there is a lot of potential which is growing for these protein-based amyloidic structures in material design. So you can imagine that on one hand, these structures are causing a lot of diseases, but on the other hand, they can be also transformed to newer materials for variety of applications. There is another very important area where uh, the most of the drugs which are driven by biotechnology means are biopharmaceuticals, because these are protein based drugs. But the tendency of a protein to form into this amyloidic fibrous structure also remains as a genetic property, as a general property of any protein. So under a lot of stresses of upstream and downstream, proteins can also fibrillate, and a protein can lose its stability. So the company has to really work very hard to remove these aggregates, otherwise they can be also toxic to the human body. So I will not go into details of the material part and the biopharmaceutical part, but try to remain focused a little bit on the disease part. And this list which I have given here will help you to appreciate that how many diseases are caused by these amyloidic structures, which are formed in, for example, in brain, in other systemic organs, and sometimes they are also localized. And the well-known, we all know, are Alzheimer's, Parkinson, and Huntington's disease. And there are many conditions such as chronic inflammatory conditions, tuberculosis, rheumatoid arthritis. In these conditions, some of the body proteins, their concentration goes very high, and due to which they start clumping with each other and start depositing in spleen, liver, kidney, etc. And some of these forms are highly localized. Okay, so they do not spread in the body, but they cause local problems such as cataract is an example. So this list is also increasing year by year. So a lot of diseases with age, they also leads to this aggregation problem in humans. In yet another very recent development, what people have found now, that it's not just proteins which can make this long fibrous structure in although some of the cases, but the diseases which are related to inborn errors of metabolism, where there is a problem, for example, in a gene which leads to abnormal protein, and that can be abnormal enzyme, so it will not catalyze a particular reaction. And the theme is that due to this, the metabolite which will be not converted to a particular product, that concentration of that metabolite will go very high in the body, such as in millimolar concentration, and I will cite one example of it later on. And due to which now these molecules will start self-assembling with each other, and once they will self-assemble, they will elongate in such a way that they will form long fibrous structures like protein form amyloidic structures, and therefore they are also supposed to be making one of the triggering factor of causing problem in inborn errors of metabolism, such as mental distortion. So in all these diseases, whether you talk about protein assembly or this metabolite assembly, the hypothesis is very simple, that protein aggregates build up gradually in the body. It takes years to build up, and which leads to pathogenesis, and therefore the best way to treat a patient or cure a patient is probably to inhibit this aggregation process or clear the burden of these aggregates from a tissue or the organ. So by considering these diseases and interest in materials as well as in biopharmaceuticals, we wish to understand the fundamental mechanisms that how these proteins or metabolites self-assemble, what are the atomic and molecular details which we can get by understanding the fundamental mechanism of aggregation, which eventually will help us to understand deposition and spreading mechanism in the cells, in the body, in the patients. And while we are understanding fundamental mechanisms, we are also keeping an eye for application. And I will cite a couple of examples in the product discovery, which either came directly from the fundamental research of aggregation process or as a side product, which is very important when we talk about products out of fundamental research. So in our laboratory, we use variety of methods. We use biophysical methods, biochemical assays, immunochemical assays. We use bacterial and cell-based assays. We use model systems such as Drosophila. We make the protein in the lab. We use chemistry, nanoscience, yeast-based assays. So in a way, we try to use multiple assays to answer a particular question in protein aggregation problems. So I will start with giving you now some glimpse of our research. I will not go into details of it, but I will highlight the fundamental discovery and the product which is originated from that particular fundamental discovery while we are doing research. So first example is on a small metabolite which is phenylalanine, which is a hydrophobic amino acid. And as I mentioned that these amino acids can build up in phenylketonuria. Phenylalanine concentration goes very high in the body due to deficiency of either phenylalanine hydroxylase or the mutated form of it. So what scientists have shown that it builds up and it forms amyloidic fibril structures in the brain of patients, and therefore can cause mental retardation in phenylketonuria patients. But the question is that how such a small molecule can do that self-assembly process to make such amyloid fibrils. So we ask this question, and then you can appreciate, I will not go into details of it, but you will see that a lot of fibrous structures in test tubes were formed by this phenylalanine in normal buffer conditions. And we use multiple methods, multiple assays to probe this, and eventually we could say that yes, phenylalanine and its hydrophobic group in the side chain, it stacks with each other in such a way that it eventually elongate and grow into amyloid-like structures. Now, to prove this, we also took the D-phenylalanine, which is isomer of L-phenylalanine, and tested whether D-phenylalanine also does the fiber formation. And we found that both L and B form the fibrous structures. However, D arrest the aggregation tendency of L-phenylalanine. Okay, so and one of the mechanisms by which these protein fibers grow in the body or in the test tube is that they form a nucleus, and on nucleus monomeric protein or phenylalanine, they will start elongating into long fibrous structures. And interestingly, what we found that this seeding mechanism of elongation is also suppressed by D-phenylalanine of L. And as we all know that L and D are part of our body, so we hypothesize now that D-phenylalanine can be a potential therapeutic molecule for phenylketonuria because D-phenylalanine and L-phenylalanine both can use the same transporter to enter the blood-brain barrier. Okay, and if one of the causes of toxicity in phenylketonuria is L-phenylalanine aggregation and fiber formation, D-phenylalanine can also use the same transporter and go inside the brain and localize in such a way with L-phenylalanine and either changes its aggregation pathway or slows down the aggregation tendency of L-phenylalanine. Okay, so this is our product one discovery as far as L-phenylalanine aggregation in phenylketonuria goes. Although we are going to see now whether it also shows this kind of effect in, let's say, animal models of phenylketonuria. Now I will switch on to another important question which we asked, that what happens if phenylalanine as an amino acid is also coupled with a hydrophobic moiety such as FMOC. FMOC is shown over here. So FMOC and phenylalanine, these derivatives of amino acids are used in peptide synthesis, and therefore we thought that we wish to see whether FMOC phenylalanine can tweak the properties of aggregation of phenylalanine, although it is not related to phenylketonuria, but we just wanted to understand the fundamental mechanism of phenylalanine influence or vice versa of the hydrophobic residue. And interestingly, what we found now that FMOC phenylalanine by itself does not form the amyloid fibers, but it forms a hydrogel. And this hydrogel we understood the fundamental mechanism of hydrogel formation. I'm not going into the science of it, that how the molecules self-assemble with each other and entrap a lot of water molecules, which we call as a hydrogel. But we immediately thought that it can have multiple applications. So we loaded this hydrogel. First of all, we established that it slowly releases the FMOC phenylalanine monomers. And then in the hydrogel, we could encapsulate different kind of dyes or drug molecules for slow release. So we thought that this gel can be utilized for slow release medium for multiple usages. But interestingly, what our student found that the hydrogelation property is also linked to critical micellar concentration. In other words, that at a typical concentration, it will form a micellar structure, and it also changes the surface tension of the buffer. And when my student tried to understand the physicochemical properties of it, it highlighted that it may disrupt the membranes. And therefore, we thought to use this FMOC phenylalanine hydrogel now for antimicrobial activities. So we started with phenylalanine in understanding side, we thought to use it for an antimicrobial agent. And when we tested it, so first of all, we developed a process. I will not go into details of it. This is a very simple process to make a hydrogel. We are scaling up it now. But at the same time, what we found that this hydrogel can act on variety of microbes, however, spares the gram-negative bacteria. It did not act on the gram-negative bacteria, but killed even MRSA. Okay, and then we tested this activity on a wound model of mice and also filed a patent. It turned out to be very, very interesting molecule as far as this gel tendency to kill microbe goes. At the same time, we found that it's not just gel. The solution form of FMOCF also kills the microbe with the same kind of action, which I am coming in a short while. And to probe the mechanism of action, what we found that the FMOC phenylalanine from the gel disintegrate, form a micellar structure, which eventually interact with the membrane, disrupt it. As you can appreciate it from this particular figure, this is a debris which is obtained from the microbe, and this is one which is untreated, and this is a control. So we did a lot of assays that how exactly it is working, but I will just shorten it to this particular diagram. So what we found that FMOC phenylalanine gel releases the monomers. At higher concentration, it will form a micellar structure, which will disrupt the membrane. But we also found very interesting property of this molecule. At low concentration, this FMOC phenylalanine seeps in to the microbe and causes a lot of stresses such as oxidative and osmotic stress. And collectively, all these mechanism leads to clearance of the bacteria. So this is product 2 from our laboratory. And as I mentioned that FMOC phenylalanine is just gram-positive specific, we wanted to make it more broad spectrum. And one of my student, Avinash, he thought to make a combinatorial approach, and he chose AZT, an antibiotic which is gram-negative specific. It impedes the final stage of bacterial membrane synthesis. So what we hypothesize that if AZT by itself little bit disturb or impede the membrane synthesis of gram-negative bacteria, it may allow now FMOC phenylalanine to act or go inside it. So that was our hypothesis, and we tested that And what we found, first of all, we of course made the co-gel of FMOCF and azithromycin, that is AZT. We successfully made. We characterized it, extensively. We studied its release kinetics, etc. And finally, what we found that FMOC phenylalanine and AZT, to cut the story short, eventually did what we hypothesized. AZT weakens the membrane, FMOCF now access itself into the bacteria and eventually bring all those stresses which we have seen in case of gram-positive bacteria as well. And the effect was synergistic. AZT and FMOCF now works at a very low concentration and also kills the gram-negative bacteria in a faster manner. So what I told you till now, that we started with phenylalanine. First of all, we discovered D-phenylalanine as a potential product for or potential molecule to treat phenylketonuria. We went on to make a FMOC phenylalanine hydrogel, which is gram-positive specific, and in combination with AZT, we also made it broad spectrum, that it kills now both gram-positive and gram-negative bacteria. Just to highlight one serendipitous discovery, because we are talking about product development here, and we should not just focus all the time on what we hypothesize. We also should look, discoveries around the shelf. And what we found here, in one of the experiments, when we were trying to understand the fundamental mechanism of FMOC-F assembly mechanism, we wanted to see whether FMOC and phenylalanine, if mixed together, can form this gel, or the covalent bond between FMOC and phenylalanine is important. So in such a reaction, when we used HPLC, we found that all these molecules arrives at different retention time, but we were also started seeing a newer peak of FMOC phenylalanine. And that also in water. We do not have to do any kind of a chemical reaction. The reaction was spontaneously happening. And this serendipitous discovery led us to find the synthesis of FMOC phenylalanine in just tap water. We do not have to use any chemicals which otherwise are used for synthesis of these amino acid derivatives. So we developed this process, and we extended this process to other FMOC derivatives of amino acid, and it came as a serendipitous discovery. And we have published this paper in Green Synthesis. So I'm not going into details of it, but we have a method now to synthesize, and probably we are going to scale it up now, the synthesis of FMOC derivatives of all the amino acids, just in tap water. So I will summarize my first theme on product discoveries on the basis of phenylalanine, where I talked about phenylalanine aggregation mechanism. I talked about how we have discovered D-phenylalanine as an inhibitor of L-phenylalanine, and then a mechanism of FMOC phenylalanine hydrogel formation, its antimicrobial property, and how we converted FMOC and AZT hydrogel as a broad spectrum antimicrobial effect. And we have also developed this green synthesis process for making FMOC amino acid. I have not talked about Himanshi, who is one of the organizers team, how this FMOC phenylalanine worked on both gram-positive and gram-negative bacteria's eradication of biofilms, and which has a lot of potential in materials as well as biomedical devices. So I hope I am audible, and still there is some time.
Yes,
Hello?
sir.
Okay.
Yes, sir. You can go ahead
Okay,
briefly, sir.
yeah, thank you. So quickly, I will go to the next half of our discovery, which we have done on Huntington's disease. So in this disease, what happens is that the protein which is responsible for aggregating in amyloid-like fibers has a mutation in the gene, which is known as Huntington gene. And when it is expressed, it leads to the incorporation of lot of polyglutamines in one of the domains of this protein. And this glutamine repeats leads to the aggregation in the brain. And eventually they form very high beta sheet rich aggregates, and eventually leads to cellular abnormalities, transcription problems, mitochondrial problems, calcium signaling problems, and neuron die, and a person develop lot of problems in this particular disease. Importantly, though, we have animal models now, cell models, protein and peptide models to recapitulate HD. So just to cut the story short, what we found now over the years that how this polyglutamine containing protein self-assemble and aggregate into these long fibrous structures. And we have been asking the question that can we find some hot spots in this aggregation pathway, which we can use now rationally for drug design. Okay, so this cartoon, which is shown here in orange and green, depicts the picture of aggregation pathway of Huntington protein in Huntington disease, and various steps in this particular stages are the targets of drug discovery. As I mentioned, we use different kind of model systems. We use polypeptides, where this QQs are mentioned of different lengths, because length has a direct link to aggregation tendency and appearance of the symptoms of Huntington's. So we have all kind of lengths of polyglutamine repeats in our laboratory. We also used neuronal cells where Huntington protein is expressed. We have yeast model system. We have also Drosophila model system of Huntington, so that whenever we wish to evaluate the efficacy of a particular discovered molecule in vitro, we can directly test it there in our laboratory and with the help of our collaborators. However, we have to develop a process, because when we get these polypeptides synthesized from a peptide synthesis company, they do not work directly. So over the years, we have learned to handle these peptides because they are very much aggregation prone. So they have to be solubilized first. And we developed a very nice process where we use TFA and HFIP. And later on, we found that TFA is more than enough to disintegrate these aggregates. And once they are disintegrated, we have incorporation of sedimentation assay in our laboratory, which we developed over the years. And we can make these proteins and polypeptides soluble for some time, so that their kinetics and thermodynamics and aggregation phases can be studied very nicely. And also when a molecule will arrest it, we can really study in laboratory conditions. So we have developed this particular process, and we have also got a patent for this. So now we are screening different kind of molecules for Huntington protein whose aggregation pathway is well established in our laboratory and in others' lab. So we are taking different approaches. Some approaches are those where the molecule is already known for, let's say, aggregation inhibition. So in that sense, arginine is a well-known hydrotroph. It inhibits, in general, the aggregation of proteins. It also assists in protein refolding. We chose another molecule, which is known as calmidazolium chloride. This molecule was screened in a high throughput assays for A beta aggregation for Alzheimer's disease, however, was never tested on Huntington. So we also chose this molecule. And what we found that arginine arrests the aggregation of Huntington protein. But to understand the structural activity relationship, we also chose multiple other molecules, such as derivative of arginine, arginine methyl ester, arginine ethyl ester. And what we found that arginine methyl ester and arginine ethyl ester, as compared to arginine, is much more better and potent inhibitor of aggregation of Huntington. And we found it later that why. whether we tested it in yeast model or whether we tested it in cellular model, whether we tested this in Drosophila model, these molecules inhibited the aggregation of Huntington protein. Arginine inhibited to some extent, and in all these assays, arginine ethyl a better fashion. And what we found, I'm just summarizing a very, very long story and making it too short for you. The mode of action, this is in between is Huntington protein. It forms oligomers and eventually form these long fibrous structures. Arginine acts by breaking the hydrogen bond network, which develops with polyglutamine interaction in Huntington protein while it is aggregating. Arginine ethyl ester, what we found, it also worked the way arginine ethyl ester work, but ethyl part uh, binds to another region which is toward the N terminus of Huntington and brings better activity of Huntington aggregation inhibition. That's what we found. And we also got a patent for these molecules. So in yet another molecule which I have mentioned, CLC, calmidazolium chloride, this is a very good case study. I feel what happens that when we were trying to understand the activity of inhibition of calmidazolium chloride on Huntington, this arrested the aggregation initially. However, uh, as the time progresses, this molecule adsorbed on the plates, whatever the material we use. Okay, so in generally what happens, we as a scientist do not really worry about adsorption of molecules on the surface of the plate, etc. If we do not see any activity, we believe that molecule is not active. But here, one of my brilliant student, Virender, look this question and look deeper into the mechanism of adsorption of calmidazolium chloride in the plates. And what he found that CLC by itself act as an inhibitor, but BSA and HSA, when used in the solution form, not on the plate by say, but in the solution form, can bring back the CLC and keep it in the solution form. Interestingly, now BSA found the complex with CLC. Okay, so CLC is inhibitor to the less extent because it is absorbing and getting lost by adsorption, and it is not available to the Huntington protein. But BSA brought it in the solution, but made a complex with CLC. And interestingly, this BSA CLC now, the figure is shown here that how CLC is interacting with the BSA or HSA, and it brings even better inhibitory activity of the Huntington protein. So we started by looking CLC, but here we are having two products in our hand. One is CLC alone and one is BSA CLC alone. Now, because Huntington disease is a brain disorder, so any molecule which we discover, they have to pass through the blood-brain barrier. And what Birinder did, he made now the nanoparticle of BSA and CLC as a therapeutic approach, so that BSA and CLC will not disintegrate, for example, in the blood, and both of them can seep through the blood-brain barrier into the brain and most likely work there for inhibitory activity of Huntington aggregation, and therefore hypothesized to treat this particular disease. Now, we tested the efficacy of these nanoparticles on neuro2 cells where Huntington protein is expressing and aggregating. We saw that the activity retains there. We also tested these nanoparticles and these complexes on Drosophila model. There also we did see the relief of symptoms which are caused by HD expression and aggregation. So,
Sir,
yes.
sir, you have 5 minutes to wrap up the
Okay,
presentation.
okay.
Sorry to interrupt.
Yeah, no problem. Yeah, thank you. Yeah. So what we got essentially is that we have aggregating protein where now CLC or BSA CLC complex or CLC loaded nanoparticles can act on suppressing the aggregation of Huntington and eventually treat, inhibit the aggregation and eventually treat the Huntington's disease. So I will skip another very important part. Here we have discovered again the mechanism-based peptides which arrested the aggregation of Huntington, and now what we are trying to achieve basically, that some of these peptides which are inhibiting the aggregation, they have to be taken to the brain, and we are in parallel developing nanoparticle based approaches where some of these peptide inhibitors are loaded into these nanoparticles, and we tested their physicochemical properties so that they can cross the blood brain barrier, and eventually also tested their efficacy in Drosophila models. And what we are finding is very impressive data, that these molecules which we are developing in our laboratory, we are making nanoparticles to take them to the brain. They are working on, at least on the model systems of Huntington disease such as Drosophila. So from the Huntington disease perspective, I have mentioned that we discovered mechanism-based inhibitors for Huntington disease. Considering polyglutamine aggregation as a target, we have series of peptides in our hand. We have arginine derivatives, we have calmidazolium colloid, and more such as osmolytes. And we wish to take these discoveries now forward. So this is the paradigm which we all are aware, that as a R&D scientist, as a professor, as PhD students, we in our laboratory discover newer products and processes, but the path is long. So we are somewhere here, I should say TRL4, but we wish to take our discoveries forward for commercialization or future developments. So I'm not talking about amyloid diagnosis because of time, but towards the end, I would like to acknowledge again CIR organizing team.
Sir, you are not audible.
Hello.
Yes, sir. Yes, now you are audible.
Yeah, so I sort of thank now everyone for listening to my talk, and if you have any questions, I would be very happy to answer. So thank you very much for giving me this opportunity.
Thank you, sir. Thank you for this wonderful talk. So I would like to take my friend Vaishali's role for a few minutes because I really want to thank Professor Kanan and Professor Thakur for joining us today. So both the sirs are an important part of my research journey. So Professor Kanan taught me when I was in BTech, and Professor Thakur really helped me to go on a research journey when I was doing my master's in IIT Kanpur. And I'm really privileged to get both these experts together on this stage today. So I really deeply thank you, sir, and thank all other presenters as well to be here and to just support us in the conference that we are taking today. Thank you, sir.
Thank you.
Thank you, Manju. Thank you, Ashwini sir. We are really glad to have you addressing all of us. Now I request Dr. Devnath Pal, Professor, Department of Computational and Data Sciences, IISc Bangalore, to deliver the first invited talk to the conference. Dr. Devnath Pal obtained his PhD degree in chemistry from Jawadhpur University in year 2000, NAD MSc from IIT Kharagpur in 1995. He is a recipient of Young Scientist Medal, Indian National Science Academy, New Delhi 2000, Alexander von Humboldt Fellowship from Germany 2000. He's also a recipient of NASI Scopus Young Scientist Award, National Academy of Science, Allahabad 2010. His research interests include computational biology, bioinformatics, genomics, proteomics, metabolomics, drug discovery, methods, and algorithms. Dr. Devnath Palsa, over to you.
Am I audible?
Yes, sir, you're audible.
Okay. Let me upload my slides first. My slides are visible?
Yes, sir.
Okay. Good afternoon to one and all. I thank the organizers for having me today for this wonderful event that they have organized for the students. So we have heard several lectures from the morning. So I believe mine is the one that is computational. So I'll go a bit slow, and if you have questions, feel free to ask, or we can have a discussion later, after the lecture as well. So the topic of my lecture today is systems medicine, steps forward. Now, as all of you know, biology is a horribly complex subject. And the reason is that it is extremely heterogeneous. There are thousands of molecules. Our body, does almost 1 trillion reactions per second. So it's not easy to, study such a system. So we have been doing studies on biology for long, and we have made several advances, great advances in medicine. But with time, we realize that, the approaches need to become holistic. The reason is that many often the, prevention is better than cure, and solutions are rather simple when interventions are done early. So for purpose of health, that is scalable and effective and personalized, systems medicine is something that is the future. And one must say that our own system of medicine that we have in India, based on Ayurveda, that is believed to be a functional systems medicine framework. But unfortunately, we have lost much information that may have been used in developing these frameworks, the molecular and biochemical information. So it would be interesting and and an urgent need to work towards this goal. And today I will try to illuminate your ideas, so that in future when you're studying biology, medicine, these concepts that we are trying to develop, not only in my lab, but many other labs in the world, you may find useful. So let's get started. So my talk outline is the first topic, the point I will cover is systems medicine, a perspective. Then we'll be bridging the scales. So how, scale is a problem in biology. Then I will walk you through 2 studies, case studies that we have done. Face today, and what, according to my opinion, is a future perspective. So let's first look at what is systems medicine going to be, or what we envisage it may look like in future, and what is to be done. So systems medicine has 4 different modules, I would say, that together will build the systems medicine architecture. One is the personalization, one is the generalization, one is delineation, and another is integration. So each of these modules have an important role in developing the systems medicine architecture. Now, if we look at the current way of therapy, so if you have a patient, if you are a patient yourself, so you go to a doctor, so the doctor checks you, he takes the readings, maybe your biochemical report, your body temperature, you have to answer questions from him. So you basically give data to him, and the physician has a prior training. So he basically analyzes the data that you give to the doctor, and a therapeutic decision is given to you. Okay, so this is currently the way things are done. So now what we want to do is that we want to give the doctor more prognosis and diagnosis information, and most importantly, we would like to give such an information which is customized for the patient. So that is the ideal situation. So that is what our goal is. Okay, so now, as of now, what we are able to do is we are collecting this data, which is essentially clinical data, and we also have molecular data from different experiments that we do in the labs, and together we are trying to come up with data mining, techniques and analysis through which we are trying to find indication, and this indication is being fed back through research papers and technical reports, which is given to the doctor. So that is what we are currently doing in most cases. But this is not a holistic way of doing things, right? So we need two other modules. One is the generalization module and the delineation module. The bioinformatic data that we are getting right now, has to be, in such a manner so that it is, caters to the population or a subgroup like a cohort, or in a smaller group like that. So the sample size should be such so that it's distributed, representative for the population that is being targeted. So we are now, we know that medical informatics is very important. So this data that we are gathering patient by patient in hospitals slowly are being fed into the system. So data management is very much necessary to gather the data and organize it in a certain way, such that we can use the data to do biostatistics and epidemiology studies. We are able to classify the data, stratify the data, and provide a justification for whatever data that we have gathered, and feed it back to the physician, so that it helps the doctor in making a decision. However, one must understand that as patient, each person is unique, and one a priori doesn't know that whether the patient who has a disease or some symptoms, whether that data is already present in the database or not. Okay, so which essentially means that whatever we do by gathering information and trying to classify it or stratify it, we need to actually mine it and develop principles and mechanisms out of it, so that it can be used in a wide manner, and this is what is called generalization. So this generalization is the critical part of the whole architecture, because this is what will allow the physician an explanation when he or she is going to provide a therapeutic decision specific to a patient. So for personalization, generalization is the most critical part. And as you can see, for doing this, we need to have in vitro model, we need to have in vivo model, we need to have clinical trials. And for this, we need two different approaches. One is of systems biology, and another is experimental molecular and cell biology. And at the heart of it is the network and PKPD modeling and simulation of whatever models we build. Okay, so this is the key part. So although we are having technologies which are very high throughput and advancing day by day, so we will, as the technology advances, we will keep on gathering data, okay, and we will be mining it. But eventually, for the success of systems biology and personalization, you would need to have develop principles and mechanistic explanations, which will actually refine the personalization approach. So in my presentation today, I will try to show you how we are trying to develop models and simulation techniques to help this process. And as you can see, once we generate principles and mechanisms, we can feed it back such that there's a feedback here, so that whatever we are doing, all these things go in an iterative process, so that eventually the quality of therapeutic decision making is improved. So that is basically what systems medicine is, and how we envisage the architecture to be in the future. So what are the key challenges that we are currently facing in systems medicine? Obviously, the density of information in a physiological system is extremely high. I already told you when I began my talk that we are, having approximately 1 trillion chemical reactions in our body every second. So, which is enormous, right? Which reactions are ongoing, which reactions are switched off, switched on, what are the reaction rates, which proteins are being synthesized, which are being inhibited, which species is being transported. And not only that, our body is in an environment. So we are also constantly exchanging, molecules from the environment. We are breathing in oxygen, we're giving out carbon dioxide. So water vapor is going out from our body. So it's not that it's an isolated system. So it is not very easy or a simple system. The information is very hard. The next challenge is the heterogeneity among the physical components of the system. So even if you just take a small molecule, as we heard in the previous talk, even you take a small molecule like phenylalanine, okay, you expect it to behave normally in a certain way, but and under certain situations it behaves differently, which means that there's heterogeneity in its its physicochemical behavior. This is a small molecule, right? So imagine you have proteins, you have tissues, so they are not entirely identical in the way their structures occur, or they behave in a given context. So this is very important. Therefore, each part of the system present challenges in modeling and analysis. So which means that the details are very important if we have to do things properly. An easier way to address the challenge is to identify which component of the system is critical for altered response and is key to reducing computational analysis and complexity, which means that if you are having a disease, you need not simulate the whole body, the whole system. You may localize your analysis or your simulation to that part of the body which is showing alteration compared to the normal system. What is also important is that even though we are gathering lots of data these days, such data will only be effective when the data is covering all parts of the solution space, right? So if we are only gathering one kind of data and we don't know that there could be other kinds of data within the solution space, which means despite the fact that we would have mined it intensively, we would not still be able to get to the solution. So these are the key challenges that we face. Now coming to bridging the scales. So normally when experimental biologists are working, they're typically working with a given protein molecule or a small molecule, or maybe they will be working with a cell system. Okay, so we would want to actually do the analysis in a more holistic manner, such that if we want to gather inferences, these inferences that are observable are typically at the level of tissues or organs or in the organisms, right? So phenotypes, when we see, these are observables at the higher order, but we typically want to understand how they originate, and these are occurring at the DNA or the RNA level. So we have to bridge the scales, right? So the computing must be such, or the conceptualization of the problem should be such that it is easy for you to go up and down this scale. So this is one of the challenges in multi-scale biology as well. So let me now come to what we are doing in our lab. Okay, so in our lab we work with many different levels of biology. These include the DNA, the transcriptome, the proteome, and the metabolome. And we essentially try to derive algorithms and methodologies to improve our understanding. We also do computational drug discovery. And when we see there's an opportunity to propose interesting leads to a given disease for which we may have a model, we also try to do that. So in the work today that I'll be presenting you, I'll try to present you two case studies. One case study is for a protein, and another is for organ transport of metabolites. So let's start with the protein molecules first. So as we all know, protein molecules are the biological workhorse. And currently we know that our body has more than 30,000 different proteins that are expressed. Okay, and proteins have a specific structure because they are made up of specific amino acids, and these amino acids join together and create certain structures in our body, and these then fold to give tertiary and quaternary structures. So these are essentially molecular machines. However, because they are chemical molecules, and because they have conformations, which means there are free rotatable bonds, so the structures can change. And because the atoms have also charges, so they also respond to the environment. So this makes it a versatile machine that can respond to different situations. And as we will see, that our current approaches to understanding these molecules are still not so great. Why am I saying this? The reason is that currently what we try to do when we try to gather function for a protein, we use what is called an evolution-based function assessment, wherein we try to find, wherein we try to align the sequences. And by virtue of aligning the sequences, we are able to understand how similar the sequence is to the subject sequence to which we get the match. And based on the match, we try to say that yes, the match is good, so it is going to have possibly an identical structure, and as well as an identical function. So based on this, we have developed concepts of proteins that are all called orthologs and paralogs, and these are actually for all the proteins which are homologs. So I'm sure, being a biotechnology student, you know what are homologs, orthologs and paralogs, and I need not explain this further. But in reality, it is possible that a given sequence can have more than one function. Okay, so while evolution can give us possibly one function or maybe more than one, there could be additional functions for that sequence that is entirely based on the physiochemical properties. So what I'm trying to say is, the knowledge of sequence or structural similarity may not be enough for functional inference, because a function is governed by a collection of different protein properties, each of which in different combination may give rise to a different function. Okay, so this is a different way to understand function, not by trying to find homologs, which is essentially matching of the gene products. And since we find that two things are matching, we also think that the functions are also same. But as we will see, that this approach, although is very popular and also powerful, has its own limitation. So to highlight that indeed the features of the proteins, the physiochemical features of the proteins are important, you must look at cases of moonlighting proteins, which presents evidence that how physiochemical features of proteins alter their function. So moonlighting proteins, as you can see, I have given several examples. So say this is a protein in this example A. The same protein, as you can see, this is a protein from E. coli, is PUTA. The function is different when it's bound to the membrane as opposed to when it is bound to the DNA. It's the same protein, but when it is bound to the DNA, it does one function. When it's bound to the membrane, it does another function. There's another case where the same protein, when it is present inside the cell, does one function, and when it goes outside the cell, does another function. There is no change in sequence, and as we know, if the sequence is identical, the expectation is that it will also have the identical structure. But you see, the environment is different, so it might have changed the structure and is performing a different function. Other example is the same protein which is expressed in different cell types. For example, here is a neuron has an epithelial cell. So the same protein, two functions in two different cell types. The other case scenario is when a given protein, for example, an enzyme, has a cofactor bound compared to when it does not have a cofactor bound. So typically, what we know currently is that when an enzyme has a cofactor bound, it will do its catalytic activity, and it is probably inactivated when there is no cofactor bound. But that need not be necessarily true. Similarly, if the protein is just monomer compared to its oligomeric form, or a protein which is just by itself, or complexes with another protein, or a protein which has multiple modules, and each of which can get activated at different points of time. So now you may be asking that, whatever I am saying currently, how it is useful in systems biology. So you may appreciate that when we want to prescribe therapy, if we don't know what is the root cause of the disease, then we would not find a good solution. So typically our idea is that we would fix certain proteins which are probably malfunctioning or which are overexpressed, or which are underexpressed need to be activated such that the disease condition is remedied. So unless we have a good idea that which protein is doing what function, the library of proteins that one may target is incomplete. So the dream of giving a personalized therapy would never be fulfilled unless we have full knowledge of the possible functions that a protein might do. So our goal is to actually develop such a method which is able to cater to all these conditions. So thinking and looking at the problem from a systems biology perspective, function can be used as a pivot for holistic inference. The reason is that if we say, if we're looking at a multi-scale problem, so which means we have a phenotype that is, say, seen at the tissue level, and we want to link it back to a domain level in the protein. So we can see that there is a link, say, from a tissue function to a cellular function to a molecular function to a domain function. Okay, if we were to make a framework based on structure, we would not be able to make such a link. So function gives us a seamless, multi-scalability when we are looking at the systems medicines perspective. And this is because function inherently is a multi-scale, activity. So which is defined as any of a group of related actions contributing to a larger action, especially the normal and specific contribution of a bodily part to the economy of a living organism. So function is not something that is exclusive. A function is always has parts. So these parts together make a given function. That function may join together with other functions to give rise to a bigger functions. So a higher level function will arise from many lower level functions, and a lower level function will be part of many higher level functions. Okay. So this gives us a good framework to approach the systems medicine problem and develop methods and algorithms. And as I've already told you, the example of enzyme. So an enzyme is expected to catalyze, but when it is not catalyzing, perhaps the cofactor is absent, or maybe there is no substrate. The protein is not sitting idle, it is involved in regulation.
Sir,
Yeah.
so sorry to interrupt, but you have 5 minutes to wrap up your presentation.
Okay. Okay, fine, no problem. So, so what we are trying to do is we would like to, find out that what regulatory activity this enzyme is doing. Fine. So let us see what we have done. So what we have done is essentially we have developed a method which is called DIN-FUNC. The DIN-FUNC is a method which maps dynamics to functions of functions in protein. And as we all know, functions of proteins usually reside in their flexible regions. Okay, and dynamics is agnostic to sequence and sensitive to the environment. Okay, so we would be able to also see the response of the protein. Dynamics can also explain the multifunctional aspect of a protein, including moonlighting. However, dynamics, the way currently it is a very compute intensive approach. So a genome scale application is needed. So in our lab, we developed a cross-grain molecular dynamics approach through development of a new force field. So for those of you who are new to molecular dynamics or may not know much, so these force fields are essentially potential energy equations. So these equations essentially govern the dynamics of the molecule under equilibrium or non-equilibrium conditions. So one needs to formulate them. And if you are able to formulate them, then you will be able to predict the motions of the protein. So we developed this approach in our lab, and a PhD student has worked on it. So what we found after we developed this approach, that if we take as loop regions of proteins, and when we find out their flexibility signatures and we match them through such a matrix, then we are able to find out through their flexibility signature or the fluctuation signature whether is a possibility of these loops participating in a common function. And to our surprise, when we did this, so this is one example. So this is a profile of two proteins which have identical function, and this match is done by a mathematical approach called autocorrelation vector profile. So when we match these profiles, you can see from this matrix, you get very high matches. So each box corresponds to a match between one frame in the molecular dynamics, and we take 11 frames. So this box has 121 cells. So we see the very high matches, and when we match against some other protein which does not have the same flexibility signatures, then there is a poor match. So this approach gives us a very powerful way of finding functions. So we have actually done large-scale validation of this method, and we have found that the true positive rate is about 87%, and the true negative rate, sorry about the typo, true negative rate is 92%. The exciting part of this is that if we look at the proteins that we are able to match the functions to, so these are proteins with extremely low match. So one protein has only 3% match, 13% match, 9% match. So these proteins, you would never get function match using common available evolutionary based techniques. But through this method, it is possible. So these are certain other examples that we found. I'll skip. There's another protein that as an example to show about moonlighting. So what is the take home here? The take home here is that flexibility of protein molecule is very important, and this flexibility also offers us a signature, and this signature, if such a stretch of the protein is involved in a certain activity, could be used to identify its function. So this is an example from an experimental NMR structure showing you that only one change in amino acid can change the whole fluctuation profile of the protein molecule. So if we are able to find out these changes, so even in disease cases when there are mutations, one can quantify whether these mutations are essentially lead to loss of function. And if there's a loss of function, we know that it could be disease causing. So this presents us with a very powerful way of finding potential markers or polymorphs of a given protein that are disease causing. So what is the summary? The summary is that if we do simple analysis of protein flexibility through molecular dynamic simulation, it is a very powerful approach to understand function, phenotype, and diseases. And since this is an era of machine learning, we could also use machine learning to improve our understanding of these flexible regions and matching the autocorrelation vectors that we use in this current algorithm. The want of time, I will not be able to cover the next part of my talk. So for those of you who are interested in this dynamics work, so there are 4 different publications that are already there. You may read it. So I'll skip this part and go to the last 2 slides and I'll finish. So what are the challenges of computing that we face when we try to develop systems medicine framework? So we need to be capable of improving our pattern recognition by integrating high dimensional data. Okay. Whenever we have lack of data or missing data, our method should be robust to it. We should also be able to handle vague and incomplete information, which is noisy data. And data-driven training and adaptation is necessary for improving the analysis based on experience. What is my future perspective? Context-dependent alteration of readout for the same input data is better handled by physics chemistry concepts due to the infinite context space. Okay, which means that you are, as a person, you're staying in, say, Delhi or Lucknow, but if you go to, say, Ladakh, where the environment is different, okay, so your physiology will change. So likewise, if you go to some other place and stay there for some time, then your physiology will also respond to that environment. So this is essentially an infinite context space. So these things are better handled through physics and chemistry concepts. We must use high dimensional data for robust systems medicine outcome, and machine learning can help significantly in filling the missing data and parameters. I think I've kept my time. Thank you, and I'd love to answer the questions. I'm done, Mangshi. I hope I kept my time.
Yes, sir. Thank you so much, sir. May I now request Dr. R.N. Baral, session chair, to deliver the vote of thanks to our speakers.
On behalf of CIR, I thank Dr. Ashwini Kumar for delivering a lecture on product discoveries originating from fundamental protein. And also it was icing on the cake that he explained how to approach this subject, bioscience and bioengineering, and how you can go for BTech, MTech studies. Also, I thank Dr. Devnath Pal for delivering a lecture on systems medicine. Incidentally, he was the first speaker to agree to our conference, and we are very, very happy that such an intelligent person and intellectual person agreed to our conference. And we thank you from the bottom of our heart, sir. Thank you so much.
My pleasure.
Thank you so much, sir. Now I would request Ashwini sir and Devnath sir to accept the token of appreciation from CIR.
Thank you.
Thank you everyone for patient hearing. We will now be starting with session 2, which is of panel discussion. May I please request Dr. R.M. Mehra sir to become the session chair for this session, and Ms. Himanshi Singh will be the panel moderator for the session. The member for the panels will be Dr. Manisha Jindal, Professor and Dean School of Medical Science and Research, Sharda University, Greater Noida. Dr. Bhaskar Bhattacharya, Professor, Mahila Mahavidyalaya, BHU, Varanasi, Uttar Pradesh. Dr. Sally Lucas, Dean School of Allied Health Sciences, Sharda University, Greater Noida. Dr. Jitendra Gupta, Assistant Professor, Department, Pharmaceutical Research, GLA University, Mathura, Uttar Pradesh. And last but not the least, Dr. Reena Gupta, Assistant Professor, Department, Pharmaceutical Research, GLA University, Mathura, Uttar Pradesh. Himanshi, you can take this forward.
Yeah, thank you, Vaishali. So a very good afternoon to everyone. I'm thankful to all the speakers for these amazing sessions. And today I have the privilege to moderate a panel conversation among amazing people and leaders from academia. They're going to share with us their perspective on future of healthcare innovations and unique opportunities they envision in life science entrepreneurial sector. My name is Himanshi and I represent Team CIR, and I feel absolute pleasure to welcome our panelists, Bhattacharya sir, Jitendra sir, Reena ma'am, Sally ma'am, and Manisha ma'am. So I hope all the panelists have joined the session. And here today we have a diverse group of amazing people, all pioneers in the field of health sciences, and also a physicist to have a unique and different perspective. So we are first going to start by hearing from each one of you. How do you see this moment of life sciences? Today, in this era of COVID, we have so many vaccine options. Still, we are compromising on clinical trials data, research data, which proves that we still do not have a very clear hold on the COVID. Similarly, we undergo so many compromises in our labs every day. So I would like to know, by putting up my first question, which is, what are the most promising opportunities that we see at the stage where the life science or healthcare sector is today? What are the most daunting challenges that prevent us from delivering all the opportunities? In summary, what are the possibilities and what are the obstacles that stand in our way? So let's just begin with Sally, ma'am. Ma'am, are you there?
Yeah, Himanshi, thank you so much for providing a platform to put forward my thoughts and processes. First of all, I think I would convey my gratitude to the entire team and congratulations to the entire team for putting up such a wonderful and thought-provoking conference. And yes, it is the need of the hour. Most importantly, let me just go into, because I'm heading a allied health area, let me just explain to you that, programs of physiotherapy, radio imaging technology, medical lab technology, OT technicians, dialysis technicians, and cardiovascular technicians, optometrists. These are the few allied health areas which are there, and they have been there quite a few from quite a few ages. The only issue, however, seemed to be that the right direction to nurture them into the to the healthcare system, be part of the mainstream healthcare system, was somewhere it was missing. Somehow, I think COVID has opened up a kind of a Pandora's box to understand that yes, these are people who are relevant and are the backbone of any healthcare systems. There have been, the possibilities of, the healthcare stakeholders are confronted with the need to provide services which are underpinned by quality elements such as safety, effectiveness, timelines, equity, efficiency, and patient-centeredness to complex patient presentations. While there have been innovative means to address these challenges, newer models of care and use of technological solutions in healthcare workforce continues to play a crucial role in helping to meet these evolving challenges. And believe me, when I say you talk about challenges of the allied health professionals, I have, lack of quality of the clear workforce structure, a lack in understanding the career progression of these people, diverse workforce participation, distribution and stress, burnout, isolation. There is kind of an identity crisis which the allied health segment.
I think we've lost the voice. Is everybody able to hear her?
Yeah, ma'am, I guess there is some network issue at Sally ma'am's applications. Sally ma'am, are you there? Sally ma'am, we are sorry, we cannot hear you. Okay, I guess Manisha ma'am, you can take the conversation from here.
Yeah, thank you, Himanshi. I would like to wish a very good afternoon to all the organizers and the member panelists. It is great to see Professor Bhattacharya here, a very eminent physiologist, Dr. Gupta, and the others. So regarding your question, Himanshi, I would just like to say that Dr. Selly is right. COVID has opened up a Pandora's box, but I think it is for the better because in some respects, it has come to light that India has the ability to take the life sciences, to take the innovations, and to merge it with technology and take it forward at a speed which is very enviable. And that is what the world has seen. So we have the systems, but as far as the next part of your question is concerned regarding the challenges, the only issue I think that is there is lack of cohesion, lack of integration, and the barriers, that is the silos which are there. Once these silos are broken, once a person can freely interact and take his idea forward and move to the next step, I think the job is done. Opportunities, of course, we have a big pool of intellectual brains in India who are going abroad and working. So it is basically the environment that makes a difference. How come our Indian scientists, our people working in life sciences here, are not able to give that kind of input which they are able to give abroad? So the concern is that environment which is required, because it helps you in maturing, it helps you in putting your thoughts forward, making them integrated with other systems, and taking basically the life science research from the lab to the bedside. And that is the need of the hour.
Right. Very rightly said, ma'am. So Bhattacharya sir, what are your views about this?
So please unmute yourself.
Yeah, thank you, Himanshi. Namaskar, Manisha madam. Good to see you after a long gap.
Yeah, sure, sir. Thank you.
My sincere thanks to Professor Mehra for roping me in, because probably I am the odd man out in this group. See, as mentioned by Professor Jindal, we know that majority of the technological development or the breakthrough that came, that we are experienced with, we were taught in our schools also, those were developed during the Second World War. It was an enormous pressure on the entire scientific community, technological community, and many things came out immediately. If we compare the same scenario of Second World War, not in that light, but in other aspect with the pandemic that we just experienced and still experiencing a little bit, the pressure that this COVID created to the scientific world, that gave many things. That let us understand that how we can work, how we can develop materials, how we can develop medical facilities, how we can grow in terms of the all aspects of life sciences together to cater the public, the general public, and the vaccine, everything. So not to blame the odd scenes, but yes, the positive aspects to be taken up. Yes, there were problems, and still there are problems, especially countries like India, where we have scarcity of funds, huge population, and therefore to cater everyone and to satisfy everyone, it is very difficult. What we need to do is channelize many things, grow step by step, look at where were the faults and what we can do. Probably we need to make a chain of many things, chain of sciences, chain of developments, chain of the term that has been coined, healthcare workers. Probably that is not limited to the allied health or the nurses, and it goes up to the experts, the doctors. So all those needs to be channelized, brought together on a single table, understand their problems, and then move ahead to develop things in much better fashion. That is my view of looking at this problem.
Right, sir, right. So channelization is very important. Like in research, it's very rightly said, the more number of people channelize their thoughts, so there are a lot of perspectives to look at. So from here, so I would like to request Jitendra Gupta sir to express his views on this particular question. Sir, you have to unmute yourself.
Good afternoon to one and all. Now, life science lends important insight into the disease processes and allow the development of the novel therapeutics and innovation medical devices, and thereby directly improving the human health. So the research necessity of the life science community to work across the diverse domain and the computer, cyber infrastructure, and data experts to leverage the opportunity in the data-enabled science in the life science result in a pressing need for the validations and the producibility of the results through the new standards and data sharing capabilities. A perceived gap between the needs of data in our life science and the current funding initiatives. A specific need to integrate data enables life science with major international and national initiatives.
Yeah, right, sir. So, Reena ma'am, would you like to add your thoughts to this?
Good afternoon, one and all. Are you getting my voice, ma'am?
Yes,
hello.
Audible. Yes.
Is I am audible?
Yes, ma'am. Yes.
Okay. So good afternoon, one and all, and thanks to give me the opportunity to just talk about the emerging topics where we can say about the life sciences. See, it's a life means everybody want to go with the future prospects of life sciences. What are the biggest challenge with pharmacy? So should I talk about in the sense of pharmaceutical or health sciences? There should be the Bhattacharya sir told like that about the challenge in the chain system. Yes, we definitely required a well-development chain system where we can supply chain disruptions. Every industry can face the same problems. So we have to identify the problems which we have to be linked with the practical purpose. What we are doing the research in that aspect where we are just giving the data, but data was not completely fulfilled by the public, and same data reproduced in the same time. So what we are thinking, there should be collection analysis of huge amount of data, and there is a need of some agility and transparency, that if we are getting a negative result, we should show the negative results instead of going to positive. Always whenever we are seeing the research PhDs, M.Pharmas, M.Tech, MSc, what the people are doing, they are always showing positive data. They are not showing negative data, and negative data is a very big challenge for the people of Indians, or we can say in industry peoples, where they have to start with the new journey and they have to give certain points where they can go better ahead with these negatives, and the negative converted into the positive. So this is the most biggest challenge for, we are saying that people are always showing if 50% negatives are coming, that's why we are lacking behind with US or something, USA or Russia, because people are doing manipulations. We should not do the manipulations. And the second thing which I am thinking regarding the industry was, what type of challenge was facing by was the clinical trials. That is a lag in R&D department. Generic companies are not showing their interest in R&D. So what happens? Whatever the data was coming, that was full-fledged people were showing in the market, and which was not correct one. People are not getting the right medications. And then there is a very good, we are saying that a global competition. There are some sectors where multinational, competitive, and particularly in the research area, companies are coming one by one, but they are not showing the right data. And if right data was coming, that is a very big, very good challenge. See, this Bharat Serum, They're preparing Covaxin, Covishield was coming to the market, and it was the right vaccine. That's why people are get benefited. And with the chain system. So this was the things which we have to be managed, and we have to think of particular health sector, or in case of life sciences, in case of computational technology, we have to be go by this way. Thanks, Amma.
Thank you, Reena ma'am. Sally ma'am, would you like to complete what you were saying?
I don't know from where you were not able to hear me, but I guess I think I did talk about the possibilities of, especially because I look into the factor of an allied health. I'm speaking purely from their perspective, and I felt that yes, there have been a lot of obstacles that they have been facing over a period of time. I was talking about distribution of stress, burnout, isolation. I was also looking into the identity crisis where an allied health professional do get into, but what, despite this, despite these diverse professions, people coming in from different strata, different, areas of allied health, they all pooling together their resources and their knowledge in bringing up the healthcare system. And that is what is more important. And I feel that, yes, there is a lot of possibilities that have opened up, especially after the COVID. And I feel that to whatever extent that the COVID may have hampered us, but yes, the healthcare system, it has given an impetus to the healthcare system, which is probably the need of the hour. Thank you.
Yes, ma'am. Just like Reena ma'am also said that the negative data is also important. As we know, the positive data is a finding. It tells you what happens. Similarly, a negative data is also a finding. It also tells us what does not happen. So according to a researcher, both of these are important. Okay, all right. So from here, we all know that life science is majorly about core research, in which we study alterations in pathway, causes of a disease, understanding some kind of abnormality. But there are rarely eureka discoveries in healthcare, eureka discoveries in life sciences, just like the great Einstein, perhaps. So how can we today train our researchers to be more inclined towards translatory research and, not towards solving up theories and things, just to complete their entire degree with one particular, solution to that particular problem? So I would like to hear the thoughts from, maybe Jitendra Gupta sir first.
My views regarding this translative research. So, if we have been any research scholar, we give the project like that should contribute or solve any social problem or any social issue. So that's why, so they can put their efforts in the research that may be beneficial and also enhance the economy of the India. Another, in regard the translative research, generally we having some basic knowledge regarding the life science that we will translate. We will after translation we will convert into such a manner after the treatment, efficacy, effectiveness, that should be ultimately maybe beneficial for the human beings. So that indicates the translated research. So that's why motivated our scholars. So they give some contribution in the improvement, or we can say means we can say that so our economy of the India may improve.
Right, sir. So Gupta sir just said that our research should be about solving a social issue. It should be about having an impact. So Manisha ma'am, what are your views on this? Ma'am, you have to unmute yourself.
Yeah. Okay. Am I audible now?
Yes, ma'am, you're audible.
Himanshi, what Dr. Gupta has said is correct, and that is what I would like to add upon, is that our scientists, they don't know what they have to do. They just know that they have to do some research and they have to show some results. What we need is basically the need to know what is to be done. Like in COVID, people knew what was to be done, so they were able to perform. I personally feel that Indians have the potential, they have the capacity, they have the capability, but they can work under pressure because the sense of direction is not there. If you give them the sense of direction, they can actually perform. That is one thing. Second thing is for translational research. Again, as I said earlier, there are some silos. So all the clinicians do not have access to lab facilities, and all the researchers working in the labs do not have an access to a clinical knowledge or the clinical needs. So again, breaking that silos and having a common pool coming together to work, and by that I mean that there should be an integration and a horizontal networking between disciplines. And I know Dr. R. M. Mehra himself has been a very strong proposer for that. That we need to horizontally integrate different disciplines together, and alongside we also need to integrate the top down and bottom up system. We don't know what the people sitting on the top want and how the people at the base level can provide that. So it needs to be an integrated, a cohesive work which has to be done. And also, in addition, I would say that not only that, the scientists or the researchers basically till date, till a few years back, maybe till a decade back, they did not know how to show that they have done something. So they were not getting credit for anything that they had done. The credit was given to only people who actually got it translated into something meaningful. So that credit, that patent filing or that IP rights, those were things that the basic researcher, researcher working in the lab had no access to or no knowledge of. So uh, having that knowledge has also strengthened the position, and I'm sure that this is going to go forward in a big way.
Right, right, ma'am. Very correctly said. So I also remember, like, from my own experience when I joined my master's, like my master's lab, and when I was a very new student for research, I was also very confused on what problem to take on, because as a student, we usually do not know where to start from. So yes, obviously our direction is important, just to begin, and a right direction will lead us to right results. So yes, so Sally ma'am, I would like you to speak up on this.
Yeah, thank you, Himanshi. I would definitely agree to what Manisha ma'am and Dr. Gupta had just mentioned. To carry on, I think for every research student, there's one question which is always put up. There's something which is normally spoken to them when they take up or start doing their presentations. That is, what is a research question? And that research question has to be such that it should be something which should be able to be taken up and is kind of be able to provide an answer to us or provide a solution to a problem that one is seeing or seeing in the society. So that part of the question itself becomes a solution, and that solution now needs to be presented in the form of a patent. It can be in the form of a product, and then the right nurturing for that particular research in form of to help them a hand-holding process in bringing it to the industry, the right clinical trials, helping them to apply that knowledge in the industry where it is required, and the target population on whom you are actually benefiting that research work, that needs to be identified with a set protocol. Now that is where we are lacking, because we lack in the initial process itself. Like, what is the research question? What are you addressing, or what your research is going to address upon? And if you're able to understand that research question, the problem that you're set to solve, then you also understand, you work out your objectives for it, you work out the hypothesis, and you know what your expected outcomes are going to be in. And that of course will you as you initially spoke about the eureka moments, They come because of these kind of research questions. We're actually solving something which the society needs it so badly at this point. Yeah, thank you.
Yes, right, ma'am. So actually, like I would like to add up on this. So actually, what happens is when we as students, we begin with one particular research question, and we see it in terms of how much time we have to complete our degree. Then we begin with that question. Then we end up with negative results. Then we modify our research question. So that whole research question becomes a gray zone for us. So at the end, there is so much of confusion, and we end up presenting all the results all together with no actual outcome. So yeah, so I would like to invite Bhaskar sir to put his thoughts on this and tell us what happens. Like, is this the same thing in physics also in his department?
Yes, absolutely,
So
absolutely, yes. It is everywhere. But I would start. I would like to start, from a little. The better word is one step back. See, when we talk about research, categorically, it has two very clear aspects. One is basic research, and the other one is the applied research. So you cannot stop either of these. You need to continue with the basic research also. You need to continue with the applied research, and both these, they have their own ball game, own field, own requirement, everything. See, if I talk, from my field of view, my expertise, then let me take one example. Say one energy device. Energy devices, they are the requirement of the day. We know even the laptop I'm using, the mobile I'm using, it contains a battery. We need to look at how we can modify, how we can have high energy density, blah, blah, blah. That is one type of research. Sounds very interesting. People run behind it. It can give money, many things. But at the same time, one need to think of the materials. One need to think of what are the materials that can be used, what is the basic principle behind all these functions, and therefore that research is parallelly important. Now, if you take one person who starts working on the material, develops a prototype, then goes to the market, tries to sell it up, it is impossible. And if one thinks of all those things added together on one single chain, then that research will neither be a material research nor be a device research. It will be something like a kitchen. So therefore, these two things need to be categorically differentiated: basic research, applied research. And yes, for applied research, they have huge requirement of the outcome of basic research. For basic research, to start with, to define the path, what type of topic one should take up, there should be some guidance from the applied research, and therefore these two must converge at a single point and then again take their own path. So that is the main requirement. You do research in basic field, applied field, be it in life science. Even people started working in microbiology in 70s, and still people do lot of research on biochemistry, microbiology, and those things. Biotechnology started somewhere in mid 80s. Still, you think of 1972, 2022, people are doing biochemistry. That is the basic research, and that is very essential. At the same time, in the last 2 years, we had seen variety of applied researchers that came up with the products. So can we stop either of them? No, we cannot stop either of them. We need to think of which one is more required for public, more required for academia, more required for industry, and then classify people, classify institutes, classify the research, and focus on all the three or all the four, whatever category we make, do research on all aspects. Yes, at one point of time, we need to make them sit together, share their expertise, share their views, and then go back to their own respective places, do further research. This is my feeling.
Yes, sir. Yes, very rightly said. The discussion is very important between the basic research and the applied research, because basic research gives you the concepts and applied research gives you the application of those particular concepts. Right. So, Reena ma'am, what do you want to say about this topic?
It was very truly said by the Bhattacharya sir that there are two sciences, basic sciences as well as this applied sciences. Should I increase some points with him also? See, the translation research is nothing. It's basically a C which produce a more meaningful and applicable results. Which the goal of this translation research is that the translation moves the basic science. Means what the basic sciences we have discovered is more, and it will give more quickly and efficiently into the practice. And if it is comes into the practice, it will gives 100% effect to the peoples, and society will get benefited from that particular translation research. Nowadays, what happens? People are taking medicines. I will start seeing 90% of students are just believing on the mobiles. They are read everything from the mobile itself, even the notes, even the class material. But because this 2 years last because of Corona, and we can say that now new technologies are coming to the market. Previously, with our age and our class library, and go and see the books, and where we can take the literature and we are reading. Nowadays, what happens? Students are not able to go to the library. They are not able to visit into the certain things where they want. They say they are wasting their time. Just they are opening the mobile, seeing on www.google.com, open the files, and just read it, and they don't have any. And that's why you are seeing nowadays all PCI makes the stringent rules for pharmacy colleges. Why? Because people are making like this only. क्या है कि ये जमाना ऐसा आ गया जहाँ पर so the things are coming like that. This was the right time where we have to think about the translative research, where we can do 100% efforts. People has to go for human trials, clinical and T3. In the translated research, we have to go with this T1, T2, and T3, make the data fruitful, which will be gives beneficial to the human as well as social life. And when we are applying this research, results were coming, people will and society will get benefited. That's why government of India, Manini Modi ji, just want that Atmanirbhar Bharat, because this Atmanirbhar Bharat, how it will prepare, because small scale business are going, people are used to trial market. Every government of India has applied so many applications for the beneficial of the public only. So he knows the things. Hindustani people knows everything, but thing is they are not able to apply the innovations. So this was the basically a link where if we go with the research in case of life sciences, we have to move toward the translated research. And if translated research will be there, we will find some databases which help us our research.
Right. Very unique thoughts, ma'am. So just like you said, the role of government is important. So I would like to take that point from there, and I would like to ask the next question, which is, how do you think Indian government should help us to perform better with our assets? Because today, if we see, we have a huge bunch of people registering as PhD students, as research scholars, or as JRFs, but there is less of innovation that we see today. So how do you think, for example, I'll give you, so person who is doing a PhD after 5 years, so there is no stipend that is given, and there are some more restrictions. So how do you think Indian government can help the students to strive towards a more innovative path during their PhD journey or during their research journey? So Sally, ma'am, we'll begin from your end.
Yeah, thank you, Himanshi. I think yes, a very valid point taken up, especially the role of the Indian government in encouraging these kind of reforms, especially in the healthcare sector. In fact, I think the COVID did present a situation where they and especially they did present a situation where healthcare system had to be brought in the forefront, especially the which Dr. Reena was talking about, the Atmanirbhar Bharat Abhiyan. which these are these packages which included several short-term and long-term measures for the healthcare system, including, there's something called as the PLI, which is production-linked incentives schemes for basically for boosting the domestic manufacturing of the pharmaceuticals and medical devices. Now, if you look into that part, I'm also looking at that, it has brought out several opportunities for India to grow, and it has opened up a floodgate of Indian startups, mainly, which has risen to the occasion and accelerated the development of low-cost, scalable, and quick solutions. And also, what this pandemic is done also, it has provided an impetus for the expansion of telemedicine and home healthcare market in the country. And these kind of initiatives by the government has definitely boosted up not only the homegrown research and the research laboratories. What it has also done is, I must mention this because of the fact that I come from the allied health part. They have also come out with the National Commission for Allied and Healthcare Professionals Act 2021, which is long time pending. And I think this is a boon for every, allied health professional, that they have now very, very specifically jotted down, brought up, and acknowledged that, there are allied healthcare professionals, and they need to be substantiated not only in their research work, but also substantiated as the core backbone of the mainstream healthcare systems. So I feel that, yes, they are doing their a lot bit, and I think it is for us to raise to the occasion and contribute to our maximum potential. Thank you.
Yes, ma'am. Actually, COVID has grabbed everyone's attention towards the importance of healthcare research, because when we face a particular problem, then only we realize, what were the points that we were leaving behind since so many years. So, Bhattacharya sir, what are your views on this? How can government help us even more to, use our assets better?
You said very correctly that last 2 years, many things opened up and we experienced practically these things which probably were not in practice. Yes, the government had opened up their variety of options for startups, for research, for variety of grants, so that quickly the COVID menace that could be addressed. That type of approach cannot continue forever. Government cannot stop everything and focus on to one problem and develop one sector. So that cannot continue forever. Now, the issue is, say, for example, if we compare with IT sector has grown for maybe last 40 years like anything. The basic reason is the number of users that vary from a kid using online platform for studies to big institutions, companies, and big things. So there are a variety of users, variety of applications, and therefore that sector has grown like it. Now the health sector has grown in last 2 years because we had something which was used, the number of users were huge, but it will not continue forever. Somehow we need to focus in this direction so that the medical condition throughout the country, the up to the individual person, poorest person, the village, if that is improved, then definitely number of users will increase. But that need to be a simplest way, not a very complicated fashion, so that a person, even for the example that was quoted by Sally madam, that the telemedicine user. So how a person can use the telemedicine, that need to be conveyed to him, and therefore there must be a handshake. There must be some handshaking between one sector to the other sector. Now, one more thing I would like to point out here, as a scientist, as a professor, I can talk about, I can think about a problem, I can address to that and prepare a prototype, but can never make it B2B or B2C type of thing. And therefore, that is something which is required. And that requirement is to be fulfilled by the funding agencies, by the government, by somebody else, not by us. So the professionals, the doctors, the allied health workers, the pharmaceuticals, all the academia, they can work on the materials, they can come up to the prototype, but to make it public usable, to make it the business to the customer, somebody else need to intervene, and that somebody else the government can do, or only big industries can, not us, the academic people. That help is very much required.
Yes, sir. Yes. So, Manisha ma'am, if you would have been the policy making authority, or you would have some authority, what would you do to make the research more innovative?
Tough question, because that kind of sums up everything. But I do believe that what is required is a good industry academia interface. And there are some agencies which have been set up. The DST has set up BIRAC, which is again an interface between industry and academia. But it needs to be more proactive. And as I said, there has to be a common pool of resources, because it is very difficult for every institution to set up a research facility right from the basic sciences to the translational side, right? So what is required to a common accessible pool in most regions, and that can be with the help of corporate funds, that can be with the help of public-private partnership, or it can be a government setup. But those pools for basic research and basic, so that has to be facilitated. Not everybody is very good in basic research, and not everybody is good in translational research. So for innovations, what is required is a good merger of life sciences, of technology, and innovation. And I think a good amalgam of these three would pave the way for sustainable development. And for that, public-private partnership, a good interactive network between basic scientists and translational scientists, and use of technology, be it for providing data or analyzing data, or be it as a source of material. See, for innovation, you need to have some background knowledge. So it could be an open source data, it could be an open data system, open science system, whatever it may be. It is required so that people know what is available, what are the gaps that are there, and then those gaps can be addressed initially at the basic level, and then it can be progressed to the translation.
Right. Yes, ma'am. So maybe Jitendra sir can add his views on this further. So please unmute.
Already Bhattacharya sir, Manisha madam, and Salim madam already discussed on this. I want to add some points. I want to add some points on it regarding this. Government already started various startup, MSME, entrepreneurship, and project fundings. Already we know government already in case of IT sector, just use we can say that Narendra Modi started teleconversations, agriculture on the phone. You can utilize it. You will get the conversation. You will get the benefits regarding agriculture. How we can enhance the productivity and crop productivity. That government already help us all these ways.
Yes, sir. There are so many policies, but there is always a scope for improvement that we all see, because there are challenges that we as a professors and we as the students, we know the challenges, and according to that, government just can revise their policies. Yes. So, Reena ma'am, you can put your views on this.
Ma'am, I will give my views. There are because we are saying that their government is giving now last 5 to 6 years, government is trying to give more and more benefit to research and innovation, because Modi ji wants India on the number 1% of efforts. So government can give the fosters innovations by 4 basic ways: by buying it, by reducing the risk, by collaborating, and making some standard regulations. So these 4 steps, if government is doing, they lead to a very good customer. And government gives variety of policies. They are giving variety of policies to encouraging the development by giving direct funding to R&D. They are increasing the tax incentives for R&D, the protection for the intellectual property, and forming the just cooperative relationship between the university and the private sector, the university and private sectors. Because there are 2 parts. One is called as industry, and another one is called as an academia. If academia people are knowing everything, they are giving a good research in terms of productivity. Their research is by the industry, and industry gives very good terms. I will tell you, there are certain good in the universities like BHU have, then Delhi universities. They are government universities. So people of the industry just want to take their product from them directly, and they give thousands and crore rupees for the particular product. And this product was again patented and come into the market. For this, I will think Orthobam. You have seen ad on the TVs. They are the products of just pharmacy or health line, where they're getting thousands of earning and give more economy to the India. So this was the role of government, to give the chain link between the academia and the industry, where they can tie up and go for the good research, and that research will come into the society.
Right, ma'am. So just when Manisha ma'am explained, so there is one question that came into my mind, because she said that collaborations are important. So today we all are talking about making the research journey shorter. How we can take our lab discoveries to the bedside of people, to the people use it every day maybe, and so there are chances that instead of doing the research as an individual, we can come together and we can just work better as a community. So Manisha ma'am, since this was coming from your thoughts, so I would want you to express your views on, do you think that good collaborations would reduce the traction time between research and commercialization, or will it increase the traction time because of?
Definitely, Himanshu, it would definitely reduce the traction time. See, if I talk about doctors, because that is what my field is, if I talk about doctors, we are actually worse than laymen where technology is concerned, where mobile service, even handling a mobile is a big thing for us. However good mobile we may buy with lots of technology, we just use it for using, or maybe for just WhatsApp. So we are very weak at computers. We are very weak at, so having, like, if AI, artificial intelligence, merges with medical sciences, and that is what has been done in our institute also. So we can actually do we can have breakthrough results also, because what used to take a long time earlier, going from one phase to the next phase, doing all the screening, compiling the result, doing it manually, all that is a thing of the past now. So if we actually collaborate, and that is what I said is interdisciplinary research. Interdisciplinary research is taking the expertise of one specialty, merging it with the expertise of the other specialty, and then so definitely it will decrease a lot of time and it will become more interesting.
Yes, ma'am. I also think the same, that how we used to, and we now also we analyze our data, roughly on the computer softwares. And if there would have been a person who is expert in computer software, that person must have given us a better result or a better implication of our data. That's very correct. So Bhattacharya sir, tell us what happens, collaborations work there, or what happens? Let us.
See, talking in practical terms, we lag behind. We lag a lot, be it in physical sciences or life sciences, everywhere. This collaboration and collaborative research with industry and academia, that is still, on the notebooks, and practically it is very rare. And this is something which needs to be extensively encouraged and done. As mentioned by Professor Jindal, see, the data analytics, the handshaking of medicos with data analytics is absolutely necessary. Now we know the words AI, big data, and machine learning, everything. These were not in our time. But now, once these things have really come up, and we have experts all around us, and unless we exchange our feeling, our requirement with them, they will not be able to know our problem. And once we mutually know that how we can help each other, we can do things much faster. I will take one very small example. In one of the conferences, I saw that dentist, he said that when we take the photograph of a tooth, then an ordinary camera that gives a lot of reflection, and the actual requirement what a dentist needs to see, that is not captured. As a physicist, as a person of science, we know how we can reduce the glare, how we can reduce the reflection, maybe some polarizer or some other filter we can think of. But I never knew that this is a trouble for a dentist. So unless we know the trouble of each other, we can never handshake. And unless we handshake, things will not move faster. So this is something which we really lag behind, and we need to, not only with industry, not only with the big people, but even mutually we need to talk about, we need to discuss, and that requires a platform like this. A platform like this where people from different academic expertise, they sit together, they can share their problems, what are the problems and how these can be addressed. DRDO scientist once said that when the aircraft, a fighter that lands on any surface, the speed is enormous and it needs to be stopped. They need some fiber net in front of the fighter. That net needs to have enough strength. It should have something which reflects, and therefore pilot see any ordinary net cannot do it. So these are very simple things that one need to address, one need to look at and handshake. There must be many similar problems with the healthcare sector also, which I do not know. But we need to handshake and we need to discuss that these are the problems. Yes, if I can do anything, or the data center can do anything, or some other people can do anything. And then once shared together, things will move faster, and it needs to move faster, much faster than even on today we are moving with that.
Yes, sir. Yes, sir. I am also from a life science background, and as far as I know, we have a lot of problems which other domains can solve, because we usually think it in one single way on how to make lives better, how to provide relief from this or how to do research from this, but less of do we think about how to do it in a shorter time. So Jitendra sir, what do you think about this? Like, can different domains or collaboration with different people help people in life science to do the research much better?
Yes.
What do you think?
Before collaboration is much more important. We definitely can reduce the traction time through the collaborations. If we consider all of these first, just like a brainstorming, and provide the value or inputs. Beyond these two, one thing is also important regarding the equal partnership. So now, if we follow all of these three, brainstorming, providing the value, and equal partnership, definitely we can reduce the transaction time. So when member a team that become collaborate, they are able to utilize the knowledge, experience is mutually beneficial for the employee as well as the organizations, and because when they work together, now we can also speed up the solutions by the collaboration. Collaboration speeds things up, it fast track process, it handled by a single individual. Now collaboration also improve increase the efficiency, also enhance the employee retentions and job satisfactions. So definitely we can reduce the transaction times through the collaborations.
Yes, sir. I would also like to add one point, which is collaborations are also very risky. There is a chance that collaboration can work wonders for you, and there can be a chance that the person you are working with may not work in the manner that you work, or there may be some problems that you guys may face. Right? How risky the collaborations are, whether we should have it, whether we should not have it. Let us know, please.
Before starting with Himanshi ma'am, I just want to tell you what is a collaboration, what a collaboration defines. A collaboration itself defined in academic research that it is an equal partnership between the two academic faculty members who are pursuing mutually interesting and they're beneficial. So if you are mutually benefiting and interesting in the research, you can go ahead. There is a positive results are always be there. You both will get benefited from this. But there are certain universities, there are certain colleges, there are certain industries where the name of the only of the fake side. They are just want to go. We cannot find any results, and they cannot find any results. What you are saying about the risk, so that will be only come on the papers, not on the particular level. So it is a better person. First thing, and second thing, collaboration should be done in such a way where we can lead to applied our concept in the existing problem and lead to the solution of in an innovative way with the particular existing problem. So if we are knowing in this way, we are knowing with our ideas, our colleagues' ideas, what is happening in India, people are not showing any interest to say something, to talk about his or her own question to someone else. See, if we both are friends, like Himanshi madam and myself, or maybe I can say Manisha madam members are there, or we can say we are in academicians. And if I need the help in case of physics, something I'm inventive and I just want to go with the collaboration with Bhattacharya sir, and I should say, sir, I am having this problem. Can you solve this problem? So what is happening? Sorry, sir, don't get on negative part, but I am just giving an example. What the people are thinking, why should I interfere with her? Why should I say something to her? How we will help us? What benefit I will get from the particular things? Always we are thinking of ourselves. So when we are just living this type of thinking, I'm giving the example. I can take any example from other outside. See, there are where I'm working in GL University. What happens here? Here we are going with the MOU. We are having more than 200 of MOUs in our universities with some foreign universities also. In 4 to 5 yearly plannings are there where we can get benefited each other in this collaboration. And definitely what happens, there is a time saving, money saving. I will just to give an example also. See, I am having FTIR, I am having UV, I am having some instrument, but the same university does not have. They will ask us to do the samples for their students for some research purpose. So what happens? They have not to purchase the instrument there itself. They will collaboration, they will get benefited from this, and we peoples, both the peoples, this university and that university, combinedly join such a way where we can go with the collaborative works, and definitely this type of collaboration is very fruitful, and that will give 100% results also. Madam, please unmute yourself.
Yeah, sorry. Yeah, so very correctly said. So collaborations are basically important as well, and there are. So what happens is people usually, think about how can I get papers, how can I get patents, whether I'll be a first author, whether I'll be a second author. So everything comes on the loop. Like people are not thinking about the impactful outcome of that particular research, and we are just thinking about the impractical things, only the monetary things on that purpose. So Sally ma'am, what do you think about this?
Yeah, Manshi, I personally, I think there's a lot said, Sama, but I personally feel there's no harm in collaborations, except that, at the end of the day, what exactly is your collaboration coming out with? Is it going to be worthwhile for a researcher to share his research or her research and then come out with something which is sustainable, which can be applied, which can be useful for the society at large? Then yes, that collaboration does have a meaning, and it is important. Coming to these, any form of biomedical research or discoveries, at the end of the day, when you look into a researcher, he just plainly he would love to do his research rather than delve into something where commercials are involved, or he has to think about how am I going to develop the capitals for it or not. But if you look into the larger aspect of it, yes, there is a strong need for interdisciplinary research along with the industry interactions, because because if I say something, you come to the initial question which you asked, where I mean, what are the outcomes of any research that you're going to take up? If that doesn't have an applicability value, it should be just about getting your PhD degree or your postdoc degree and staying with it, right? But at the end of the day, you also mentioned about taking your research work from the lab to the bedside. To do something like that, you need to have the right people in place to bring out your research with, to let them take it to that bedside. So who, as a researcher, would it be possible for me to take it at my own level or not? No, I don't think that's possible. For me, I might be knowing the nitty-gritty details of getting that research completed. But if you ask me, the investments, they might be lacking capitals to do that, and large scale manufacturing of the product that I have made, that may not be possible at my end. The market or distribution of the product, who are going to do that? And to do something like that, I need the intervention of an industry there. I would need it in the form of either an industry per se, or maybe a pharmaceutical research based company which would have to help me with that particular segment of my work. And that is where I feel, you have to be very clear in what is the product, or what is your research going to come out with, and what is the kind of research, and where exactly is it going to be applicable. Now, if you are not, as a researcher, if you don't understand that, then of course there would be too many people around you to take opportunities of your research, and you as a researcher may lack an insight into what is happening into your research. So it's important that you collaborate, but you collaborate it at a very specific segment. You should know where your research is progressing. You should know which exact industry is going to help me in promoting my product or my invention. And most importantly, I think there should not be, it's my personal view, you should not be having too many people as collaborators with you, which might actually, the old saying which goes, too many cooks spoil the broth. So I mean, you need to be very selective in your process, and that is where I feel you can have your senior researchers, you shouldn't have a proper supervisor, a guide to help you through this process of doing research, and then finally categorizing it into the mainstream market, so that your product is utilized for the healthcare industry by and large. Thank you.
Yes, ma'am. Finding right kind of collaborators is tough, but it is definitely important. As we all know that solo we can go fast, but together we can go far and in much lesser time. Also, I think that collaborations today, people think of academia collaborations, but academia and industry collaborations are something that are very new and are something that can give us the breakthroughs. Also, thank you everyone. It was a very good discussion on the current stage of research in life sciences, and the take home message from this, the research is going on at its pace, and we have all our assets. We have PhD students, like yearly around 30K, 30,000, even more than that, students enroll for a PhD program. And the only need that we have to do, the only thing that we have to do is we have to channelize everything in a very better manner, in terms of students, in terms of collaborations, in terms of what are our research problems and what should be the research outcomes. So I sincerely thank all the panelists for sharing your aspirations and views. That was so thought-provoking. I would like to invite Mehra sir to present the vote of thanks to our esteemed panelists and the audience.
Good afternoon, everyone, and the speakers. I thank all the speakers for their very nice and informative presentations. And really, they have tackled the subject in a very clear and simple way. I'm sure that all the participants must have got the right kind of message, about healthcare. I am thankful and grateful to all the panelists that they have been with us for all the time. The session was going on right from the morning, 9:30 or so. Dr. Manisha Jindal and Dr. Shali Lucas and Professor Bhattacharya and Dr. Reena Gupta. Any conference or any activity of such kind, becomes most valuable if there are speakers, they speak on different topics, but then it is very essential, and not all the participants hear the or listen to the presentations very carefully, because their interest may be different and other things. But then, if there is a system to conclude, and the best system to conclude is have panelists, and panelists, they are well reputed panelists, experienced, long experience with research and other things. So that way, around 1 hour or nearly 1.5 hours on, I thank all the organizers that they have kept such a time for the panel discussion also. So the panelist has very clearly, summarized, and the thoughts which have been generated and expressed by the speakers, or eminent speakers, experienced speakers, that it now I can feel that all the participants must have understood what really is this conference was. So I thank all the organizers once again, and Himanshi, you are wonderful. Because what should I answer if there is not a good question? How can I answer? So your formation of questions are so nice that it becomes a very good direction to the panelists to answer that yes, what is being asked. So in making a good summary of anything, the questionnaire has a very important role. And Himanshi, you're wonderful. I have experienced when you recorded my video lecture also on research. So Shweta ji, I congratulate Shweta ji, Shweta madam, that she has such wonderful people with her in her team. You and Vaishali, and then Rudra and Ayush. Four of you, just four of you, in an organization, they can do so wonders. So I thank once again all the participants, and I express my vote of thanks to the panelists and the speakers. Thank you very much.
Thank you,
Thank
Mayra,
you, sir. Thank you.
for humble words. I request all the panelists to accept our vote of appreciation from the team CIR. We really apologize, but there has been some technical glitch. We'll be sending the certificates to you personally. Sorry. With this, we come to an end of the conference, and we hope that you all had an insightful session. On the behalf of Team CIR, I would like to thank all the speakers for giving your time and delivering such an insightful and informative session. Thank you everyone for joining us and being a patient hearing and making this conference a big success once again. Thank you, everyone.
Thank you. Thank you all.
Thank you, Vaishali, and thank you, Himanshi. Thank you, sir. Mera sir.
Thank you.
Thank you, Manshi.
Thank you all.
Thanks to all of you.
Mera sir,